UPR alumni, Gallitos y Jerezanas, there at this institution, which are Marimar Benitez de la Vega. Is she around, Marimar? I don't see her. But she'll join us. And Miguel Miranda Roman. Is Miguel there? Well, he'll join us later. And so they will be part of the recruitment initiatives after the talk from Dr. Boire. So today, let me introduce you to Adrienne. Adrienne is a physician scientist at the Memorial Sloan Kettering Cancer Center in New York. After undergraduate work in biology at Macalester College, she completed her PhD in biochemistry at Tufts University in 2004 and under the mentorship of Athan Koliopoulos. She then earned her MD and then her PhD at Tufts, and then she continued to do her MD at the University of Chicago Pritzker School of Medicine in 2008, followed by a residency in neurology at Columbia Presbyterian Hospital and a fellowship in neuro-oncology at Memorial Sloan Kettering Cancer Center, I should say. After completing a postdoctoral fellowship under the guidelines of Joan Massaguet, she joined the Human Oncogenesis and Pathology program at this institution as an assistant member since 2017. And as a neuro-oncologist, Dr. Boire cares for patients with metastasis to the central nervous system and conducts clinical trials targeting these tumors. As a scientist, she runs a laboratory-based research program focused on metastasis to the central nervous system, both Lepton Meninges and parenchymal metastasis. The laboratory employs multiple complementary approaches to identify the target cell adaptations to the challenging microenvironmental constraints posed by the central nervous system.
I never really thought about what happened.
And so we are very honored to have her here today to talk about her scientific life and also the wonderful research she's conducting at her institution. So Adrienne, it's a pleasure. Thank you so much. And I have to say, and I'll take this as a privilege, and today celebrating the Nobel laureates in chemistry, two women, biochemists and microbiologists, received this honor. So for us, women in science, this is quite a wonderful day, and what a wonderful time to once again also see wonderful women scientists like Dr. Boire presenting. So here you go.
Thank you very much. Gosh, so actually, I thank you very much for that introduction. I threw in a few slides at the beginning, sort of in honor of that, because I thought about myself when I was an undergraduate and how I never really saw anyone doing what I'm doing right now. And I would have never imagined that I would end up where I am doing what I'm doing. So I thought I'd give just a brief historical introduction. So first of all, I was born in California, and then my parents ended up in Minnesota, where I went to college. And I zoomed back and forth across the country a few times because you never know where you might end up. So in college, I thought that I would be a diplomat. I know it's hard to believe, but there it was. And when I was in a biochemistry lecture, I fell in love with the TCA cycle. I still really love metabolism. It's gorgeous. It's beautiful. And after the lecture, I asked my professor if he knew how awesome this thing was that he was studying. And then he said, people literally never say this to me, and people like you go to graduate school. And so I thought about that, but it seemed kind of crazy, honestly. So I didn't go directly to graduate school. I actually became a technician for a year. I worked in a laboratory studying vitamin K, working on one of these in HPLC for a year. I can tell you it was not thrilling, but I really liked the biochemistry. I actually enjoyed the thought process, and I really liked learning how the HPLC worked. And so with that in mind, I ended up going to graduate school. And in graduate school, I worked under the guidance of Ethan Kouliopoulos, and I studied the protease activated receptor, which is a really gorgeous receptor. It's a protein that sits on the surface of a cell. And when the extracellular portion of the receptor is cut by an enzyme, that new N-terminus of the receptor kind of runs down and intramolecularly activates the receptor. So it's a receptor that carries its own ligand, which is really cool. I thought it was really cool, and I still do. And so I found actually a new enzyme that cut the receptor in a new place that we hadn't thought of before. And so my graduate work ended up being relatively high profile. In addition to that, during this process, I discovered that I really also liked taking care of people, and I liked hanging out with physician scientists. And this is where I say I really had never met a physician scientist during my upbringing. My parents didn't go to college. I had never seen anything like this life before. But what I knew when I met my advisor and when I met some other physician scientists, I thought, these are my people. This is what I want to do. So then I did the relatively crazy thing of going to medical school after my PhD. I went to the University of Chicago, and it's this beautiful campus. It's kind of intimidatingly beautiful. I ate lunch in this. This is a lunchroom, for goodness sakes. I learned a ton. I had a lot of fun. I also had a baby. So I bring in these aspects of my personal life in order for you to understand that it's possible to have a personal life and also have a life in science. After in the University of Chicago, I learned a lot about the brain, and I learned that I really loved studying the brain and that I wanted to be a neurologist in terms of my clinical work. And so we moved to New York, and I had a lot of fun learning neurology at Columbia. This is my daughter during the Columbia University years. So I had a toddler during residency. It's possible. And you can work in a big, ancient, stuffy building and still have a lot of fun. And then after my residency, I came to Sloan Kettering to do neuro-oncology training and to do a postdoctoral fellowship with Joan Massaguet. So my postdoc, I was first assigned, as postdocs are, to work with another postdoc, Ching Chen here, and we were studying how cancer cells grow in the brain. We had known from previous work in Joan's lab that cancer cells were interacting with different parts of the brain, and that these interactions with the brain were allowing the cancer cells to grow in this sort of hostile environment. And other postdocs before us had looked at different aspects of these cancer cell interactions with the brain. And so Jean was starting to look at how the cancer cells were interacting with astrocytes. So she was trying to finish up her postdoc, and I was there to help. And while we were working together, we started looking at these gap junctions between cancer cells and the astrocytes. And we started looking at the ways that the gap junctions might allow cancer cells to communicate with the astrocytes. And we thought about different ways to repurpose drugs to perturb this interaction. When all was said and done, we ended up with a really nice paper. And then also I ended up designing a clinical trial based on this. So when it was time for me to decide what I wanted to do for my own work, I looked at my interactions with patients. And that's really the story that I'm going to talk to you about today. So in my work as a neuro-oncologist, I study metastasis. So I study the spread of cancer from other sites in the body to the brain and to the central nervous system. And one of the sites of metastasis that I study is leptomeningeal metastasis. So the lepton meninges, so that's what we're referring to with leptomeningeal. The lepton meninges are the coverings of the brain and the spinal cord. So here's a drawing of what this might look like in a kind of a typical looking kind of human. So you can see here's the brain and the spinal cord, and you can see these little blue lines and plaques of disease. These are the deposits of cancer on the surface of this person's brain and spinal cord. If when we diagnose this, we do an MRI scan or we collect spinal fluid. We actually have to do both. And if you can see here by MRI scan, you can see these plaques of disease, and you can tell that it's white areas of disease here, these little claws that are sitting on the surface of the cerebellum. That's cancer that's sitting there. And this line here, this is also cancer that's sitting on the surface of the brain. When we do a lumbar puncture or a spinal tap to collect spinal fluid, we take a look at this fluid under the microscope, and normally spinal fluid doesn't really have any cells. But in the case of lepton meningeal metastasis, we see that the spinal fluid becomes filled with cells. And we see cancer cells, and we see cancer cells that are kind of accompanied by a host of lymphocytes and macrophage. So many different kinds of tumors can end up in the spinal fluid. Most commonly, it's breast or lung cancer, melanoma, or lymphoma. Regardless of what we do, and we're pretty creative and aggressive at Sloan Kettering, the overall survival of these patients is very, very low. It's only about 3.5 months on average. And this is why I decided to study lepton meningeal metastasis. We don't really have any satisfying treatments for lepton meningeal metastasis. And I care for these patients, and I care for them very deeply, and I want to help them. So one of the things that we do in order to be able to understand this problem is we sort of take advantage of the very small silver lining of lepton meningeal metastasis. So it's a horrible complication of cancer, but there is a very small kind of benefit, and that's this collection of spinal fluid. So patients with lepton meningeal metastasis, we collect their spinal fluid every few weeks. We collect it for clinical purposes. It allows us to be able to determine if the tumors are responding to our treatments, and it allows us to see if the pressure is too high in this space, et cetera. So we're constantly collecting this spinal fluid. So it occurred to me that we could use these routine collections of spinal fluid and blood as a way of understanding the biology of this disease. And so that's what my laboratory does. We take samples of spinal fluid and plasma from the patients. We fractionate them, meaning we separate them into cellular and acellular fractions. And then when we have these fractions, we're able to perform kind of a host of omics approaches. So we're able to do RNA sequencing, bulk RNA sequencing, single cell. We're able to do DNA exome sequencing on the cell-free portion. We're also able to look at proteomics or metabolomics. We can do a whole host of omics. This approach really only lets us know the molecular what. It really tells us, it lets us take apart this system and see what's in there, but it doesn't really tell us how these individual molecular players might all assemble, right? It doesn't tell us how they function. It just sort of tells us who's there. So to figure out the molecular what, we have to turn to mouse models. So we make mouse models in my laboratory, and I'll tell you a little bit more about those. And we use these mouse models to be able to take these identified molecular players and to be able to decide which one of them are the most important, and then also how they might interact together. And this really allows us to be able to construct a pathway that can explain how cancer cell might grow in the spinal fluid. And part of how we're able to do this is not just through the technological feats of all of these omics, but we really need to use a lot of modern computational processes to be able to manage these very, very large data sets. So this is one of the first things that we were able to do in my laboratory was to be able to do just such an approach. So this is a project that we carried out in collaboration with many people, and I'm going to try and shout out all the people who helped us on the way. And one of the first and most important collaborators we had was my colleague Dana Peer and Linus Mazudis. So Dana Peer is a computational biologist, and she specializes in single cell technologies like single cell RNA sequencing. So if we look again at that cytospin, if we look at the spinal fluid that we put under the microscope, we can see that there are many different types of cells in our system. And if we look at kind of an old school technology like a Coulter counter or just counting by hand, we can see monocytes and lymphocytes, neutrophils, and cancer cells. And there are only really about 10% of these cells are cancer cells. So the cancer cells are really a minor player in this population. So we knew that if we wanted to carry out single cell RNA sequencing on this system, we were going to have to make sure that we were accurately representing what we were seeing with our own eyes. So we worked carefully with Linus to perfect our sequencing and processing pipelines in order to be able to reproduce these proportions accurately. And so this is what we came up with. So this is single cell RNA sequencing here in C from 5 patients with untreated leptomeningeal metastasis. So 2 of the patients had breast cancer and 3 of them had lung cancer, and there are about 15,000 cells. So each one of these little circles is a cell in space. Cells that are similar to each other in terms of their transcriptome will cluster together in space and make clumps like this. So, and the color coding allows us, we designed the color coding scheme based on the transcriptome of the cells to be able to identify what kind of cell it is. So you can see here in these orange and gold colors, we can see cancer cells. In the red and the pink, we see macrophage and monocytes. And the green here, we see the T cells, okay? So what we have now is we have a map of the transcriptome of each cell in the system from 5 different patients. And that is a lot of data. So we had to ask ourselves, how are we going to manage this data? What are we going to look at? So we thought about this, and we thought about the environment of the spinal fluid. So if we look here in A, you can see here is the composition of the spinal fluid. So that's this CSF, and the composition of the serum, or the liquid parts of the blood. You can see here that there are many aspects that are similar, like the osmolarity, the pH is kind of similar, but then it kind of falls apart. You see that the oxygen saturation is actually quite low. The glucose is a little bit low. But when you look to see what's really different between the two, you'll notice that iron is very different between CSF and serum. The extracellular iron is very, very low. And we thought to ourselves, you know, if cancer cells have to accept living in this space, they're going to have to cope with a lot of different selective pressures. And the strongest selective pressure, we thought, might be lack of iron. So we first said, what's the null hypothesis? Well, the null hypothesis is that cancer cells don't care about iron, that fiddle-dee-dee, they see the spinal fluid has no iron and they don't really care. So the first thing that we did is we looked at the iron deficiency hormone hepcidin. So hepcidin is released from cells when cells are iron deficient. So we looked at spinal fluid from the patients and we measured hepcidin levels. And so you can see here, patients who don't have cancer in the spinal fluid had lower levels of hepcidin than those who did. And that told us that the cells in the spinal fluid were iron deficient, that those cells needed iron. So then we looked at the usual ways that cells access iron, the way that cells are able to grab iron from the extracellular space. And one of those molecules is called transferrin. And when we looked at all of these known ways, the kind of typical ways that cells grab iron from the spinal or from their environment, they were all the same between these two groups, which meant that there was something kind of cool happening. And that is what this postdoc of mine, Yudan Chi, who's marvelous, that is exactly what she decided to study. So Yudan queried this data set, and she asked what genes that are important for cancer cell, for iron binding and iron transport, which of those genes are present in this system? And so that's what this is. So if you look here in A, each one of these teeny tiny little bars is a cell. And the cells are all organized by the type of cells that they are. So there's the dendritic cells and the macrophage and the monocytes and the cancer cells, T cells, etc. Okay. When you see a red bar next to a gene, that tells you that that cell type had that was expressing that gene. So for example, we see here LRP1, and you can see that LRP1 is expressed by macrophage and monocyte population, but isn't expressed by cancer cells. Fair enough. So then we asked, of all of these genes that are associated with iron binding and transport, which of these genes are expressed only in the cancer cells? And we found only 2 of them, lipocalin-2 or LCN2 and SLC22A17. It turns out that lipocalin-2 is an iron binding protein, and SLC22A17 is a lipocalin receptor. So we thought this was very exciting. We double checked our work, and you can see this as a violin plot. So here are all of the patients here, A, B, C, D. These yellow violins represent the cancer cell lipocalin-2 expression. And the same thing is true here of the SLC22A17. And you can see that the lymphoid and the myeloid cells, those cells aren't really expressing these genes at all. So what is lipocalin-2? Because clearly it's a very interesting gene and a very interesting gene product, and we need to figure it out. So others have studied this, this gene in the past. So lipocalin-2 was originally called neutrophil gelatinase associated lipocalin, or NGAL. And it's a secreted transporter. So you can see here's a beta barrel protein, and it binds iron that's complexed to a siderophore. And it's made, generally speaking, by neutrophils or macrophage in an inflammatory process. And it's thought, because it can bind iron with a very, very high affinity, so I've included the KDs here for you, because it can bind iron with such a high affinity, the thought is that perhaps it's functioning as part of nutritional immunity, that it's allowing these immune cells to be able to grab iron away from pathogens and sort of to be able to outcompete pathogens for iron, which to us sounded like a pretty good mechanism. And so we kind of wondered if something like that might be happening in the spinal fluid. So before we got too excited with all of our kind of fancy RNA-seq data, we took a step back and we looked at old school protein expression, because before you go from RNA, you want to take a step and look at protein. I'm a big fan of protein. So first we looked at an autopsy series from some of my patients who had passed away from lepton meninges metastasis. And you can see here's the surface of the brain. You can see this red area, the lipocalin-2 positive cells are the cytokeratin positive cells. So these are the cancer cells. Cytokeratin is not present in the brain normally. These IBA1 positive cells, so these are IBA1R macrophage and monocytes. And you see that those monocyte macrophage population do not stain for lipocalin-2. So earlier I had told you that cancer cells sit on the surface of the brain in lepton meninges metastasis, and they're also floating in the spinal fluid. And you could imagine that perhaps those two populations might be behaving differently. So we checked, and you can see this is the spinal fluid from the same patient. You can see that the cytokeratin positive cell is the lipocalin-2 positive cell, and the IBA1 positive cell is the lipocalin-2 negative cell. And so we quantified all these data, and then we did another set of experiments, this time doing flow cytometry of a different set of patients, showing that those pan-cytokeratin positive cells, the cancer cells, were really the majority of the lipocalin-2 positive population. So now we know that lipocalin-2 is important in this system. It seems to be upregulated by the cancer cells. So what's it doing? So then we need to turn to our mouse friends for this. So then the first step is to ask ourselves, are the mice accurately representing what we're seeing in human disease? So we collected spinal fluid from the patients, and we were able to quantify the amount of lipocalin-2 in this system. Thankfully, there are ELISAs that exist for this, so it's quite straightforward. And the patients and spinal fluid collected from patients with lepton meninges metastasis have higher levels of lipocalin-2, as you can see here. When we collect spinal fluid from mice with lepton meninges metastasis, we can see that that spinal fluid also has higher levels of lipocalin-2. If we take a look at the composition of the spinal fluid in the mice, looking at by multicolor flow cytometry, you can see that there are, again, cancer cells and neutrophils, myeloid cells and lymphocytes, just like humans. And you can see that lipocalin-2 sits on the surface of the brain in plaques of disease, just like in humans. We took the opportunity, because mice really allow us to cast a little bit of a wider net in certain ways. We looked to see about kind of all of the different lipocalin-2 transporters that exist. So we know that there are 3, SLC22A17, which you'll remember from before, MC4R, and LRP2. But it turned out that only SLC22A17 was expressed by the cancer cells in our system. So both of our players, lipocalin-2 and SLC22A17, are present in the mice. So then we can play. So the first thing that we did was we knocked down lipocalin-2 expression in our system using some short hairpin RNAs. And you can see here, if you knock down lipocalin-2 expression, the cancer cells do not grow as well in these mice. So our cancer cells carry a reporter, a luciferase reporter, so that allows them to generate a photon of light. And this is a really nice way for us to non-invasively image the mice. So you can see here in the top, when you see this glowing area, that's an area where the cancer is growing. And here we don't see any of that glow, and we know that the cancer is not growing in that space. If we watch the mice, we can see that the control mice will die relatively quickly, But the mice whose cancer is not expressing lipocalin-2, those mice will die less quickly. We then took a look at SLC22A17, reasoning that if SLC22A17 and lipocalin-2 are working together, if we knock down SLC22A17, that should phenocopy what we see in the lipocalin-2 knockdown, and it did. You can see here, if we knock down SLC22A17, again, we reduce the burden of disease, and we have a survival benefit for the mice. But what about the reverse experiment? What if we take cells that are not habituated or don't have the capacity to live in the spinal fluid, and if we force those cells to express lipocalin-2, maybe that will make those cells more likely to grow? And that was the case here. So we have our cells that don't generally grow in the Lepton Meninges here, and you can see a little spot of cancer here and here. But if we overexpress lipocalin-2, we see robust cancer cell growth in the leptomeningeal space, and that really causes the mice to die rather quickly. Okay, so we know that lipocalin-2 and SLC22A17 are important for this process, and they seem to almost be sufficient for the process. They kind of have the necessary insufficiency that you learn about in school. So we next asked, so what's turning these things on? It seemed sort of odd to us that these markers that are, these genes that are associated with inflammation would be turned on in this system. So we took advantage of our single cell RNA-seq data set, and we asked, how inflammatory is this space? What's happening in here? And we found a pro-inflammatory signature in the macrophage, which was very important. It tells us that these macrophages are not kind of anti-inflammatory. These macrophages are not trying to quiet down inflammation. They're actually trying to cause more inflammation. And we found evidence for this by looking at different cytokines. We see elevated levels of IL-6 and IL-8 and IL-1 beta. And we wondered about these macrophages. We wondered if these macrophages were turning on lipocalin-2 in the cancer cells. So to study this, we took a look at macrophage from our mouse models. We collected macrophage from the spleen or from the spinal fluid, and we used these macrophages to generate conditioned media to treat our cancer cells. We found that that it was really only the macrophage from the spinal fluid, from mice that had cancer, that was able to force expression of lipocalin-2. And we found that the macrophage themselves, so this is a co-culture experiment over here, that these macrophages themselves weren't generating the lipocalin, that it was really just the cancer cells. So, of course, these macrophages are making quite a soup of all different kinds of cytokines. And we thought, let's figure out which ones are important. So to do this, we took some of the spinal fluid from patients that had leptomeningeal metastasis, and then we immunodepleted different cytokines. So you can see here, we immunodepleted out interferon gamma, IL-6, IL-8. There are more. But it was really predominantly IL-6 and IL-8 that seemed to be responsible for the majority of this phenotype. And so then we did the same thing again, where after we had knocked down the activity by immunodepleting, we then added back recombinant IL-6 and IL-8 and found that that was enough to help us force expression. So this altogether lets us kind of start to conceptualize a pathway where the inflammatory cytokines from macrophage are turning on lipocalin-2 expression in the cancer cells. And then we thought, why would you turn on lipocalin-2 besides iron? Is there anything else happening? And so I asked the computational biologist to see if there were any other pathways that were being co-regulated along with the iron pathways. And to my surprise, and then when I realized it, I realized that it was really very obvious. It was actually hypoxia. The hypoxia pathways were co-regulated really very neatly with iron transport pathways. And so you can see here in A, here are the single cell RNA-seq data sets. This time we're looking at hypoxia signatures here on the x-axis and iron transport signatures on the y-axis. The color scheme is the same as before. So the golden colors are the cancer cells. And you can see that they're really sampling this entire transcriptomic space to be able to solve this problem. So we thought, cool, maybe the cancer cells are using lipocalin also to help cope with this hypoxia thing, because as you remember, spinal fluid doesn't really have so much oxygen, right? So we thought, how are we going to measure this? The spinal fluid space in mice is very, very small and irritatingly small. The spinal fluid in mice, it's really only about 40 microliters. So we kind of decided to use a genetic approach. We created a reporter system, a dual reporter system, where we had an inducible reporter of NLeuc that was going to, or an inducible reporter downstream of a hypoxia inducible element. So when a cell encounters a hypoxic environment, it will turn on NLeuc and will be able to release a photon of light. The same cancer cell will have a constitutive reporter, FLeuc, meaning that as long as the cell is happy in dividing, it will continue to make FLeuc. So we have these two different systems. And when we put the cancer cell into the mouse, we're able to see how is it feeling in terms of its hypoxic environment and how is it feeling in terms of division. Is it able to grow? So we found that the cancer cells live, when you put them into the spinal fluid, they immediately enter a hypoxic space, as you can see here. But despite this hypoxia, these guys can still grow, which is really very cool when you think about it. We wondered what would happen if we knocked down lipocalin-2. And so to be able to answer that question, we went into a hypoxic tissue culture system here in H&I. And when we knocked down lipocalin-2, we found that that inhibited the ability of these cells to grow under hypoxic conditions. And we found evidence of much more, more apoptosis in these cells, really suggesting that lipocalin-2 is allowing these cells to grow in a hypoxic environment and not die. So what about the whole iron story? I mean, that was what we started with. So in order to answer that question, we had to collaborate with the Spitalnik and Thomas labs up at Columbia. Because what we really wanted to know was very clearly and very simply, does lipocalin-2 allow cancer cells to bring iron into their cell bodies? And so we needed someone who was adept at dealing with iron and iron concentrations at a very, very small level, at the femtogram level. And so to do this, we need somebody with expertise in mass spectrometry, and that's what the Spitalnik and Thomas labs are able to do. So what we did is we knocked down SLIC22A17 and lipocalin-2, and we found that in doing so, that inhibited the accumulation of iron in the cell bodies of these cancer cells. We're able to rescue it by either expressing lipocalin-2 again, which was nice, or by overexpressing transferrin. And remember, transferrin is sort of the canonical kind of boring textbook answer for how you grab iron into the cell body. So we took these cells in a dish and we repeated the whole experiment, this time in a mouse. And we found the same thing, of course, when we knocked down lipocalin-2, that inhibited cell growth in the mice, just like it did before. But this time we rescued it with transferrin and found that, in fact, we were able to rescue quite a bit of this activity just with transferrin, really suggesting that a large part of this activity, a large part of the function of lipocalin-2 is to bring iron into the cell bodies of the cancer cells. So we wondered about the other iron-loving cell type in this system. So macrophage need iron as much as cancer cells do. In fact, macrophage probably need iron more. Macrophage need iron in order to be able to perform, to be able to generate a respiratory burst, to be able to phagocytose. And so we wondered, you know, if macrophage don't have lipocalin-2, what's happening to their function? How are they surviving in the spinal fluid? What's going on? So we asked the Spitalnik labs again and Thomas labs again to measure iron in these cells. And we found that just simply the presence of leptomeningeal metastasis, simply putting these macrophage in the spinal fluid of a mouse that has leptomeningeal metastasis, will reduce the amount of iron in the macrophage. If we inhibit the expression of lipocalin-2 in the cancer cells, that's enough to replete the iron in the macrophage, really suggesting that it's the cancer cells that are stealing away the iron from the macrophage. We wondered if this mattered to the macrophage. You know, it's sort of, you know, this number is of course significant, but it's hard to know what it means functionally. And so to address that, we took a look at functional assays of phagocytosis and respiratory burst in these macrophage. And we found that this really did matter to the macrophage, that the macrophage had impaired phagocytosis in the presence of leptomeningeal metastasis, but we could reverse that if we knocked out lipocalin-2. And the macrophage respiratory burst similarly was impaired in the presence of leptomeningeal metastasis, but could be reversed if you knock down lipocalin-2. So taking all of these observations and many more together, we kind of put together a pathway where we thought, okay, so in the spinal fluid, cancer cells are encountering a macrophage that is secreting a bunch of inflammatory mediators such as IL-6 and IL-8. And that these inflammatory mediators, these cytokines, are going to turn on lipocalin-2 and SLC22A17 in the cancer cell. And that this is going to allow the cancer cell to outcompete macrophage for iron because there's just not very much iron there. And this is becoming kind of a limiting reagent for these cancer cells. They really need it. In doing so, it allows the cancer cells to divide and grow, as you can see here, sitting on the brain and causing all kinds of trouble. And it empties the bellies of the macrophage, that these macrophages now are not able to gather as much iron as they might need. So it seemed as though iron was very much a limiting reagent in this system. And we thought, what happens if we just get rid of everyone's iron? What if we chelate all of the iron in the spinal fluid in this process? So there are a number of kind of old school compounds that exist that are chelators that we occasionally use in clinical medicine. One of them is called desferoxamine, and we've abbreviated here DFO. Another one is penicillamine. So penicillamine chelates copper, meaning it binds copper and takes it away from biological processes. And desferoxamine chelates iron and binds it away from biological processes. So we treated the mice with intrathecal desferoxamine or penicillamine, either early in the course of the mouse's disease or late in the course of the mouse's disease. And you can see here that mice that are treated just with vehicle don't do well at all, these poor guys. And same thing is the case for the mice that are treated with penicillamine. However, the mice that are treated with desferoxamine do very, very well, even if we treat them late in the course of their disease. And they do have a survival benefit, so the mice tend to live longer. This suggested to us that this might be a novel way for us to be treating cancer in this space. I'm really pleased to say that this has allowed us to start working on a phase 0, phase 1 trial that will be opening in the next few months for my patients. So we're really taking an observation that started with patient material. So we started looking at cells from these patients' spinal fluid. We went to the mice and we figured out what was happening in terms of the mechanism, and now we're bringing all of that back to the patients. So I wanted to end with some of my unsolicited advice that I hope that you gain from my presentation. So I think it's really important to pay attention to what you love. Pay attention to the lectures that you just can't get enough of. I loved metabolism and biochemistry, and I still do. So that was probably a good career move on my part. Pay attention to the books that you would read if nobody knew that you had to read them. And if you're at all interested in taking care of patients, pay attention to the patients that you find most compelling. These are all questions that are going to be they're going to generate questions that are worth asking. And I think it's important to sort of keep your antennae up. I also think you should dream really big. I think it's really easy to say, well, I'm just going to do something small. But I mean, I'm working on a disease that we don't know very much about. We have no treatments whatsoever. And because of that, there are very few people in this space, and I am free to play. So sometimes dreaming big and making audacious plans really works out well for you. And I think we all know this quote, but I really love the idea of making no little plans. That you should make big plans and you should aim high in hope and work. Because the work itself becomes beautiful. And also, I did not share with you in this presentation all of the rejection and criticism that I received over the course of my training. First of all, that would be a multi-day presentation as opposed to all of the good things that happened, which is just sort of 40 minutes. Okay, so rejection and criticism happen a lot, and determination and hard work are as important, if not more so, than talent. And so don't let people discourage you. You sort of have to get a little bit Teflonized, get a patch of Teflon on your soul and just go for it. And finally, I'd like to leave you with this thing from Ghostbusters. So I love Ghostbusters. They're some kind of wacky scientists, and I guess I'm a child of the 80s. Right, so the Ghostbusters are going in and they're gonna try and capture ghosts, which they've never done before, and they don't know what they're doing, and they've got a lot of really useless things like this camera, this like 35mm camera. And so one of them wisely says, all right, well, the usual stuff isn't working. And then they come up with a plan, and the plan is bogus and doesn't work. But I think it's really important to stop and think, hey, the usual stuff isn't working, so try something new. And with that, I want to thank my lab and the people who've done all this work. So here's my laboratory. I'd also like to thank my collaborators, who I thanked along the way, and all the funding sources that make it possible for us to do the kind of bold and fun work that we do. But most importantly, I want to thank my patients and their families. And my lab really considers them their patients also. So they donate their tissues to us, and they also donate their time. They come in for extra clinic visits to see me because I'm worried about how they're doing. They come to lab meeting, they come and see how we're doing, and they're really involved in our process, and I'm immensely grateful to them. So thank you.
Thank you. Thank you, Adrienne. It was a wonderful presentation, wonderful talk, and also quite informative. I learned a lot, actually. Quite a lot, and I really appreciate your time and sharing this with the students and our participants. So we have a couple questions for you.
Of course.
The first one is it known how much LCN2 expression is in the cell lines of HER2 or HER2 plus 2? Mm-hmm.
So that's a great question. So patients with HER2 positive breast cancer are definitely at a higher risk for brain metastasis and lepton meningeal metastasis. So lipocalin-2 is not expressed at a higher level in the cell lines. And, but if we look at the primary tumors from patients who go on to develop lepton meningeal metastasis, those patients tend to have higher levels of lipocalin-2 expression. So it does seem that there is a bit of a that the cell needs to express lipocalin-2, complement, and a few other genes in order to have kind of all of the necessary equipment it needs to live in the spinal fluid. And if your tumor happens to express that earlier in the course of your disease, you're more likely to develop this site of metastasis.
Okay. And there's another question. It says, why is there such a difference in the iron concentration between the serum and the spinal fluid?
Oh, that's an awesome question too. So I have thought a lot about this because it kind of, you wonder, why would we bother making spinal fluids so sparse, right? So I collect a lot of old textbooks because it's just a habit. So a lot of the old people who were working on, a lot of the old guys studied this and asked about it and thought about the same thing. And there are many hypotheses, and the most durable hypothesis seems to be that this is a great way to prevent microbial growth. So if you create a soup that the brain sits in that is very metabolically kind of not supportive for cell growth, that's going to keep bacterial growth down. That's not really testable, is it, right? But it's fun. And you could potentially, add more, metabolites into the spinal fluid to see if that accelerates the growth of meningeal pathogens. I'm not sure, but it's a very cool idea. Okay.
And then there's another question. It says, what kind of spectrometry did you use
in order? So there are a couple of different kinds that we ended up using. So it depended on what we were measuring. So my colleagues at Columbia, they measure iron and iron ions. So they're using, they're of course looking at ions. Other mass spec that we did for this project, we were looking at proteins. And so we were doing LC mass spec, really, treating samples with proteases, making fragments, and then breaking up those fragments. And then a third kind that we use in my laboratory is where we're looking at metabolomics. And so then, of course, we don't need to use proteolytic cleavage because we're looking at a different area of chemical space. So mass spec is really just, it's one of tool of many, but you have to know where you are in chemical space in order to find the right kind of mass spec.
Okay. Then there's another question. It says, was melanotransferrin among the iron-related genes that you studied?
It wasn't. It was expressed in one of the patients, cells, but not in all of those cells. But I did start to look at all of the transferrins because I started wondering, well, if lipocalin is one answer, how do cells solve this problem in other ways, like other similar ways? The reason that we stuck with lipocalin too is that was sort of the consensus opinion amongst all of the cells, that all the cells really thought we're kind of double down on this lipocalin plan. So we thought, all right, we'll go with that.
Okay. I have a question because I noticed that there was a major expression at the cerebellum. Is there a reason why you see that more so than in other structures, like more in the cortex or the hippocampus?
So, okay, the short answer is I don't know, but I have good ideas. So one is that when you think about the flow dynamics of spinal fluid over the brain, the smooth parts of the brain tend to be places where the spinal fluid is probably going to flow more quickly, and they're probably going to be more laminar flow and fewer eddies. And so the cells might not slow down quite as much. And if you think about the cerebellum, it's really kind of rugated. And so that perhaps those are places where it's more likely that cancer cells might kind of tumble and slow down and be able to stick, sort of in analogy to the way that platelets stick. The other possibility is that the surface covering for the brain and the spinal cord has a different embryonic origin depending on if you're in the front of the brain or the back of the brain or the spinal cord. Of course. So it's possible that the surface markers and the surface molecule or the PIA in the back of the brain and the front are different, and perhaps, perhaps for instance, breast cancer cells, perhaps they like sitting, they have the right lock for that key. I'm not really sure, but it's a very fun problem.
Yes, and it's very interesting. And so I imagine the phenotype on behavior and motoric effects throughout the cancer.
Absolutely.
I imagine that those are the premier side effects that you will see in the patients.
Absolutely. So because we're talking about the cerebellum, we have the patients tend to have difficulty with coordination, but also within the cerebellum, one of the major ventricles in that space has an area next to it called the area postrema, which is the vomiting center of the brain, which, and so my patients very often have terrible nausea because the cancer cells are sort of pushing right on that area postrema, which is awful.
Yes, of course. Well, I think, there's another question. I think this is the last one before although we could be questions all afternoon, but we should move forward to these other things. But there's the last question. How are iron deficient women affected by metastasis diagnosis?
Oh gosh, yes. So whoever you are, you're like reviewer 3. So we thought we tried our best to answer that question. It's a really complex question. So it turns out cancer itself can make you anemic. So there's something called the anemia of chronic disease, which I will nerd out briefly and tell you that it turns out that chronic interferon gamma stimulation causes the ferritin molecules in your body to switch from being a predominantly light chain to a predominantly heavy chain, and therefore they scoop up iron more quickly, and a bunch of other things happen in this whole gamish to make your body under chronic immune stimulation become anemic, and makes it much more likely that you are going to have low iron levels regardless of whether or not you have metastasis. So when we tried to deconvolute this question, we had the devil's own time. We could not answer that question because you can imagine that if we were right, patients who are anemic, maybe they would have some sort of protective effect, right? You'd think, it's probably a good deal to be anemic if you have cancer. But yeah, we don't have evidence for that. I wish we did. It would be so satisfying, wouldn't it?
Yes, of course. And then another one, I can't just the students are. That is the part of these seminars to have.
It's the best part. Q&A is the best part of any seminar, honestly. It's my favorite part.
So could the lowering of iron concentration in the cerebrospinal fluid as a therapy negatively affect other cells like neurons?
So that's actually part of the reason that we're doing this as an early phase trial, because desferoxamine has never been delivered intrathecally, so it's never been given as this route of delivery. We don't really know. we do know that the spinal fluid space and the interior of the brain, the parenchyma, are two distinct anatomic compartments. And so communication between those compartments isn't always clear. That said, there's a lot of new cool neuroanatomy right now, suggesting that perhaps spinal fluid can kind of wash through some aspects of the perivascular spaces in the brain. And if so, iron chelation might chelate iron within the brain itself. And we don't really know. We didn't see that in the mice, but of course, mice are only being treated for a few weeks, and they have such small brains. So it's a difference of scale that's actually quite important. So we're going to start slowly with patients and do quite a lot of formal pharmacokinetic and pharmacodynamic work.
Excellent. Thank you, Adrienne, for a wonderful presentation and also answering all our questions. So now we move toward the recruitment part, which is as important. And for that, I'm going to leave Jahaira Fortis to introduce everyone and then start the presentation.
Thank you so much, Dr. Maldonado-Blar, and thank you, Adrienne. It's such a pleasure to see you present outside MSK as well. You almost got me to do a postdoc at your lab. Almost. Anyway, hi everyone. I'm Jahaira Fortis Santiago, and I'm representing the GSK team here along with my colleagues. And it's my pleasure to introduce you to the stars of the show here. First, we have Mike Overholzer, who is the Dean of the Gerstner Sloan Kettering Biomedical School. We also have Miguel Miranda Roman and Lepton Meninges, both of who are alumni of the University of Puerto Rico in Rio Piedras, and you are going to get to hear from them and their experiences doing research with us. We also have our assistant dean here, Linda Burnley, who is sitting there in the back. Linda, thanks for waving. And I don't want to take a lot of time from them because they are going to be telling you all about our school. So I actually want to pass the baton to Mike. Mike, are you unmuted? Yes?
I think
Share your screen, or do you want me to do it from my computer?
I can share it if it works. I think I hit present now, right? And we'll see if we can get it.
Yes.
Let's see. You see my screen?
Yes, I can see your screen.
Great.
Perfect.
Okay, so I've lost the ability to see all of you. So feel free to interrupt me, Yahara, or anyone if you want me to stop for any questions or anything, okay?
Perfect. I think with that, Mike, I encourage the students to put the questions in the chat, and I can interrupt Mike as I see fit.
Okay, great. So really the stars of the show are our students. So Miguel and Mari, you can wave again really quickly. My job is really to get us to the point where you can hear from two of our amazing graduate students at Gerstner Sloan Kettering. And then the other stars are all of you. We want to hear from you and take any of your questions and have a dialogue about graduate school, about GSK, and about anything that's on your mind. So it's really my pleasure to be here. My name is Mike Overholzer. I'm the Dean of Gerstner Sloan Kettering Graduate School of Biomedical Sciences. For short, we go by GSK. We're an innovative graduate school, and we're embedded right directly within a world-renowned cancer center, Memorial Sloan Cancer Center, right in the heart of New York City, on the Upper East Side of Manhattan. We are different in several ways compared to other schools you may look at for your PhD training. We offer personalized training to individual students. And one of the main reasons we can do that is we have very small class sizes. You can see a photo there of us teaching in the first year curriculum. And our small size not only allows for a very nice classroom experience, but also one-on-one mentoring with all of our faculty is shown at the bottom. That's Luis Parada and one of our students, Yu Jung Chen, at a microscope. And so we're small. We recruit only 10 to 12 students per year in each class. And so what that means is in total, right now we have 79 students in our entire school. We do that on purpose because that small student body has the chance to interface with a much larger number of faculty. So we're actually up to 130 and growing. So the students that we bring in each year have lots of opportunities to be mentored by our faculty and by leadership. And they have a lot of chances to do research at our institution. So as you might imagine, because we're embedded in a cancer hospital, Memorial Sloan Kettering, a world-famous hospital and research center, a lot of our research centers around what we call cancer science. But we study cancer through many different research disciplines. In fact, all the disciplines that you'd be familiar with, from basic science all the way through to translational science, and laboratories with physician scientists such as Adrienne Boire, who you just heard from. I also have a lab. My lab is in cell biology, which is one of our 10 different departments that we have at MSK, in which GSK students can explore research opportunities. And so you'll see on this list many different traditional departments: developmental biology, in addition to cell biology, or immunology, structural biology, and molecular biology. And we also have specialized programs such as Human Oncology and Pathogenesis program, which is down there at the bottom right. Adrienne's lab is actually in this department that we call HOP. And I think all of these scientists, if not nearly all, are physician scientists who, as you just heard from Adrienne, they develop research projects in their laboratories that in many cases directly feed into new clinical trials that we run right in our cancer hospital at MSK. So as a student in our graduate school, you would have all of these different opportunities to explore science depending on your interests. And we get outstanding students come to our school. As I mentioned, we're a small size. We're also a new school. We just recruited our first class in 2006. Which actually might seem like a long time ago to you, but in the lens of graduate schools, that's a very young school. And we've graduated only 75 students so far with PhDs. So out of the current students and those who've graduated, we've already published almost 400 student-driven research papers, which is outstanding. And our students have been recognized for their efforts by earning already 51 different fellowships during their time as PhD students in GSK. In addition to the research opportunities that are excellent at our institution, we have a really innovative curriculum in GSK. I'm not going to spend time going through the details here, but you can find every detail about that first-year curriculum on our webpage. And that's really the point of emphasis. Most or all of the classes you would take are in the first year. We want you to focus on coursework in 1 year while you're doing laboratory rotations, and then we want you in the lab, like in Dr. Boire's lab, to go discover big new things that relate to basic science or cancer science or all of the above. In that first year, our courses are separate from laboratory rotations. So you can do from 2 rotations up to as many as 4 to try out different labs. And when you're rotating, you're not in class. And when you're in class, you're not rotating. That lets you focus on the lab when you're in lab and on class when you're in class. Each topic in our innovative curriculum is taught by an expert in that field. Adrienne Boire would teach you exactly about what she just talked about: metastasis and how cancer cells survive in spinal fluid. Craig Thompson, who's also the president of Sloan Kettering, comes and teaches the first-year class about metabolism. So the core course is taught by more than 80 of our 129 and growing research faculty. They come in and teach you exactly what they know the best, the research topics with which in which they're making innovative discoveries right in their laboratories. So in addition to the research and the curriculum, there are a lot of other ways that we support student development at GSK, and I'll point out a few other aspects of our school. We have a competitive research stipend. This year our stipend is $42,230 to be exact, and it increases each year. We offer our students financial support to travel to conferences every year, so that you can go out in the world and show off all the great science that you're doing in GSK and in MSK laboratories. We offer relocation costs. When you first move to the school, we can reimburse travel expenses. Every one of our students who comes to GSK is supplied with a computer. We have excellent health benefits, full medical benefits, and dental. And I mentioned we're on the Upper East Side of Manhattan. We offer subsidized housing rates. So when that's combined with a competitive stipend, our students do quite well during their years at GSK. We also have a collaborative mentoring network. And so as a student, once you choose your thesis mentor among the 130 and growing options of different laboratories, not only do you have a mentoring network within your lab and with your mentor and your thesis committee, you also have me. I'm the dean of the school, and I interact with each student constantly, and Linda Burnley, shown to the right there, our associate dean. And we have an assistant dean, Tom McGaldy, who I think is not logged on today, but you would see the three of us all the time. And in collaboration with Yahara, who's also here today and shown there on the bottom left, our Associate Director of Diversity and Inclusion, as well as Thaliana and Ushma. Thaliana runs an Office of Career Development. We take developing your next career steps after your PhD very seriously, and we have a lot of efforts centered around career development in our school. And Ushma Neal is our Vice President of Scientific Education and Training. Really, the six of us provide a collaborative mentoring network that supports each student through their development as they progress toward earning their PhD. What do our students value most about GSK? Here are a few things that we hear all the time. The exceptional research opportunities at Memorial Sloan Kettering. Our innovative curriculum. Check it out online. I'm not going to show you the details here today, but we're really proud of our curriculum. We think it is the best way to spend 1 year learning how to make new discoveries that you're going to then go do in your own research, in your hands, in the laboratory that you choose. Our small class sizes, compared to other schools that you may look at, I think GSK will have the smallest sizes in your class that you will find. That allows us to have personalized mentoring. Our students value their voice in shaping our school. And they value support and mentoring, as I just mentioned, from their mentors in laboratories all the way through to leadership in the school, including myself and Linda, who's here today, and Yahara. Speaking of their voices in the school and the fact that I'm here to get you to the real stars, I want to introduce one of our current students, Mari Mar, who's going to tell you a little bit about her journey to GSK and what she's doing at GSK. Mari.
Hi everyone, how are you doing? I hope well. I'm going to tell you a little bit about my journey. So I was an undergrad at the University of Puerto Rico, Rio Piedras, like you guys, and I was also part of the Rise and Mark program. And I did my undergrad research at Dr. Grivenel's lab, and then I also was part of 3 summer programs. And then I also spent a couple of years with an NIH diversity supplement doing some research at UCSF. And then finally, I joined GSK because of all the great opportunities our dean mentioned. But the 2 things that attracted me the most were the small class size and the fact that, you know, I could only take 1 year of class, but also be able to rotate at a different at that the rotations were separate from the classes, and that, you know, that by the program being so small, you know everybody, and so your opportunities to network are stronger. And also we have the Tri-I community. We have Will Cornell and also Rockefeller University next to us, so it's very easy to collaborate with researchers there. And yeah, so I'm gonna tell you, I'm only a second year, so I haven't like pin out a thesis project, but that's what I'm doing right now. So I'm gonna tell you a little bit about the lab I joined. I joined Dr. Andrew Koff lab, and we study cell cycle regulation, specifically the senescent state. So when cells exit the cell cycle, they can either exit the cell cycle reversibly, and they become quiescence, or they go into a further state called senescent, which is irreversible. They can no longer enter the cell cycle, and this state can be characterized by different markers that are there, SA, beta gal, SAF, ATRX, foci, and SAFs. But also what is very important about this state is that it causes inflammation. And also that as people age, they accumulate these senescent cells that cause these inflammatory signals, and this is correlated to many diseases that come with age, like diabetes, Alzheimer, and cancer. So also, our lab and many others have been interested in understanding this transition from quiescence into senescence, and also it's important to study it because we would like to keep developing drugs in the context of cancer that could possibly help maintain cells in this senescent state where they can no longer keep dividing. So right now I am, well, next year, so right now I'm preparing for my qualifying exam, as most people know it. So right now I'm writing the thesis proposal. So, but I will be happy to answer any questions about the lab. We have great summer research opportunities here. We have a summer program that if you guys are interested, most PIs participate in that. So that is a very good option. And also answer any questions about the program. But yeah, thank you.
Mari, thank you so much. I'll just point out, so we're all going to be here for a Q&A panel in just a few minutes, but I'll point out this is a great example of how we think about cancer science at our institution, because really studying senescence is a basic question in science. How do cells make fate decisions? And those fate decisions are important for cancer, but also inflammatory conditions and aging. And so you can imagine Mari's project really has a lot of different potential therapeutic implications, one of which is cancer, and many of which are other pathologies that have patients that have diseases other than cancer. So that's really how we think about the discovery space at our institution. We study basic science in many cases with the idea that if it can be translated into cancer, we then do that very, very quickly. But our research may also lead towards therapeutics for other types of conditions. So Mari, thank you so much. And really the next star of the show, I'll get out of his way, is Miguel. Miguel, the floor is yours.
Hi, buenas noches. So I'm Miguel. So I'm currently in the process of starting, or in the process of my sixth year in the graduate school. So you can see is that I'm from Bayamon, and I entered the UP in 2010, and I did different summer internships at Einstein and at UPenn. When I was in the UP, I did research in the Centro Comprensivo del Cáncer with Dr. Pablo Vivas, and I applied to graduate schools in the 2015. I can tell you in a moment why I decided to join and apply to the Gersten Sloan Kettering Graduate School. But before that, I just want to also very quickly show that once I entered the school not only provides you all the support that the dean already mentioned, the school also has different, they manage and provide different fellowships that are provided to the first year students once they are transitioning towards the second year. So I was fortunate to get the support also from the school to cover my second year of the graduate school with the Palestine Fellowship. And last year I got a Department of Defense fellowship. So, and I'm currently doing research also in a lab that is actually next door to Adrienne's lab. In the laboratory of Pingxi. But can you go briefly to the previous slide? Sorry.
Yeah, no, sorry.
So before I talk about the research, I just want to, I have this in here to show you that all of you or most of you are in the stage when you are in the UPR, in the UP. And you can see what I have been able to accomplish in these past years And this is exactly what you can also do, as Adrienne was saying in her presentation. So you need to dream big, and you need to work hard, and you need to put you in the best position to then be able to achieve your dreams. So think what you want to do in your next years, what you want to do for grad school, or whatever you want to do in once you finish in your undergrad, and you need to work hard and put you in the position to be able to achieve that. So right now you are in the UPR, in the UP. So for those that are, I just want to briefly say something. For those that are interested in continuing in research, for example, for our schools likes applicants that have a lot of research experience, and also research related experiences, like maybe a presentation in which you present your research. So I just want to mention that you can take advantage of the ASBMB symposium that is going to happen at the end of this month or next month in the UP. And the deadline for the abstract submission is actually next week, October 15. So I strongly suggest you to participate in this symposium that Marimar and I were the ones that organized and started the symposium 7 years ago. So we are very happy that the symposium is still happening. So take advantage of that and present your work and put that in your CV. So you can go to next slide. So very briefly, because this is not the point of what I'm talking, I just want to talk about mostly about the school. But my research, I work also in a sort of translational project. I work in a cancer called MPNST. It's a rare type of soft tissue sarcoma. And as you can see on the left, there's no current treatment. The best treatment option is surgery, which is not achievable many times. So what I'm doing on my research is trying to develop a way that we can do a drug strategy that we can use to be able to better treat this cancer, this type of soft tissue sarcoma. And because in the clinic, there's been different strategies that have been used to treat the cancer, but this cancer can become resistant to these single drug treatments. So I'm trying to find a way to overcome the ability of this cancer to become resistant. And I already have a good strategy that we are testing, and we expect it to very soon start clinical trials. And I mentioned this is sort of translational research, but also in Sloan Kettering, we can do a lot of basic research. And maybe this is not as basic as some people would think, but also in my project, I'm working in epigenetics because this cancer has loss of the epigenetic complex PRC2. So I'm trying to see how the loss of this epigenetic complex can maybe affect the ability of this cancer to become resistant to the drug treatments that are being used in the clinic. So I'm studying the mechanisms of how PRC2 loss may be important for the development of this cancer. So I just want to reiterate, if that's a correct word, I just want to say again that in MSK, in the graduate school, when I was applying to this graduate school, I thought that we'd have a lot of translational research, a lot of translational labs. But in reality, as the Dean Mike Oberholzer showed in the presentation, we have a lot of departments that do a lot of basic science, from developmental biology to structural biology to pharmacology, immunology. We have a little bit of everything. So if doing translational research is not what you are interested on, we still may have some type of research or that is what you are looking for. So I would recommend also you to consider us, look in our webpage to see if you can find something that you are interested on. And that will be all for now.
Miguel, thank you. Thank you so much. And I apologize if you hear my dog barking in the background. I think it's almost over. But Miguel, thank you. So just a couple more minutes from me. I want to show you a picture of our students. These are not only the stars of the show today, but the stars of our school, right? Our school is for our student community. And these are some pictures at a retreat we take really every other year at a resort in New York, in upstate New York. where you have a chance to relax a little bit, and we'd like to talk about science. And on the top picture there is one of our recent graduates, actually, Zeta Zhang, who was talking about his research in a chalk talk format. He's at a whiteboard with a marker, just talking about ideas and experiments with our community of students and faculty at this really fun retreat. We have active student community groups. So we have a GSK Women in Science group that's very active called GWIZ. We have a very active student council who also plans other activities throughout the year for our student body, including a 2-day ski trip and other events. And so please go to our webpage and check that out. And I want to remind you that we are in New York City, as you probably know, but it's such a great place to live and such a great place to do science. So we're located on the Upper East Side. You can see the East River here on the left side of this photo. And that tall building with the orange color top there is the home of our graduate school. Right on the Upper East Side of Manhattan. It's called the Zuckerman Research Center building. That's the home of the GSK Graduate School. It's also the home of roughly half of our laboratories at Memorial Sloan Kettering. The other half are just down the street, 1 block. And you can see what it's like to do science here within our building. The amazing backdrops that you can get in study spaces. I think you could see out of Dr. Boire's window as she was speaking. So just a few other pieces of information that I want to make sure that you know about GSK. We have no application fee. So one of the themes that I want you to take from today is please apply. It won't cost you anything. Our application is online. It is open. What we require in the application is similar or the same as every other graduate school. A personal statement, 3 letters of recommendation, official transcripts, and our GRE is optional. You do not need to take or give or submit GRE scores. There is absolutely no penalty for not submitting GRE scores. If you want to submit GRE scores, you're welcome to do so. And the TOEFL, we have the TOEFL and the GRE and other aspects of our process included on our webpage as frequently asked questions, but I want to emphasize that TOEFL is not required for students who have a bachelor's or master's degree from an institution in the United States or in a US territory. Our application is open and our deadline is December 1st, so please apply. And I want to point out this as well. Mari touched on it. We have great opportunities for summer undergraduate research at Memorial Sloan Kettering as well. So if you're not ready yet to apply to graduate school, if you're not graduating soon, think about our summer undergraduate research programs. We have 6 of them at Memorial Sloan Kettering, and they are outstanding. One of these, called SERP there, Summer Undergraduate Research Program, is run right through GSK. So, but yet all the others are also Memorial Sloan Kettering programs. And you can see summer, SERP has a broader scope. So any of our laboratories, similar to our graduate school, are open for SERP, and others have specialized focus. For example, chemical biology, computational biology, engineering, and so on. I'll just show you a quick photo here from one of our summers. Unfortunately, in this last summer, as you're probably aware, we couldn't have students on site. I don't think anybody else could. But we were able to hold an online summer program instead. But this is a picture from the year before when we still had our on-site program, which we hope to have next summer, from the GSK summer program called SERP. So please check those out if you're interested in undergraduate research. And if you're ready for applying to graduate school, please apply. I hope you could see from today, our main priority at GSK is our students. Our priority is you. Training the next generation of leaders in science. You are all going to grow up and make the next big discoveries. I can't echo strongly enough what Adrienne Boire said and what Miguel emphasized. Dream big. There are huge discoveries out there to find. You are going to find them. And our priority is you. We want you to apply to our school, and we want you to be excited about the research opportunities that we have at our institution. So please consider joining us at GSK. And we've left enough time to take any questions that you may have with our panel. Thank you so much for being here today and allowing us the opportunity to tell you a little bit about our school.
Excellent. Thank you, Mike. We have received a few questions, and I think the first one I wanted to direct that to Adrienne. They are inquiring if you have any suggestion about making a decision to go either to PhD or MD school.
Oh, well, I think you have to ask yourself, what what do you love best? And I love science, I have to say. I mean, it's the first training that I had, and I'm very much a scientist physician and not really a physician scientist. I really love biochemistry. When I think, I'm not kidding when I say, like, when you ask me, what do I want to read? When I'm not going to pick up a medical journal. I end up reading basic science because it's more fun to me. My patients I adore, but part of the reason that I love spinal fluid is because you can easily put it on a mass spec. So for me, it's very simple. Like, I went with my gut, and I love science, and I don't look back on that at all. I think that the other thing that you should do is really ask yourself how much exposure you've had to all of these things. So it's never a bad idea to try it out, to follow clinicians around and see if you can kind of see what their life is like. See what the day-to-day life of a scientist is, see what the day-to-day life of a clinician is. I will say that the kind of clinician that I am is very much it's a very rarefied kind of clinician, right? I have a very narrow scope of patients that I treat. They're patients that have the disease that I study. And so it's very much in like the research realm. I'm not, a general practice doctor. I don't, my daughter has strep throat and I'd bring her to the pediatrician. So I guess that's what I would say.
Thank you. We have a question, Mike, asking about the summer programs. If the summer programs are limited to freshmen, seniors, and juniors, or if seniors could apply to the summer programs?
So seniors, we do not, the summer programs don't take seniors, right? So once you've graduated, you're no longer eligible to be an undergraduate in the summer program. So if you are a junior, sophomore, freshman, so on, that would be eligible. Seniors, unfortunately, are not eligible because by the time you'd come on, you would have graduated. If you're going to be staying on as an undergraduate past that year, you'd be eligible. But if you're graduating prior to it starting, then you're not eligible.
Thank you. I was going to emphasize that part. Some of the students at the University of Puerto Rico take 5 years to graduate instead of 4.
Yeah.
So if you are going for that year 5, on our scale, you will be considered a senior. But in theory, because you're still not graduating, you are welcome to apply to the summer programs in that year.
Yeah, so the key is once you've graduated, you're no longer eligible.
There is a question about, does the program have bioengineering related programs? So I am assuming it's a GSK program. Do you offer bioengineer focus?
So we haven't, it's for the PhD or the undergraduate. I'm not sure which.
I think it's a PhD, but we could answer both.
I'll answer both. So we have an engineering summer program at MSK. And so for anyone who's considering an undergraduate program in an engineering discipline, please check out the webpage for our summer programs. We have an engineering focused summer program that we share with Weill Cornell, which was mentioned by Mari, is right across the street from us there in Manhattan. So we also have, therefore, lots of labs that basically do engineering. They do it from a cancer-focused context, largely. But we have numerous labs that do physics and imaging-based approaches involving physics for both clinical imaging and also for research-based imaging. We also have really engineers. I'll give you an example, is Dan Heller. You can look up Dan Heller, is H-E-L-L-E-R. He's basically an engineer who makes devices at MSK and comes up with strategies for therapeutics. And we have numerous labs like that. So whoever asked that, if you have interest in engineering laboratories, feel free to reach out to me. I'd be happy to Zoom with you and talk about individual labs. We do not yet have an engineering curriculum for a PhD, but we have research opportunities in engineering. If our curriculum in GSK was of appeal, the research opportunities for an engineer would also would match.
Thank you, Mike. And I think either Marimar or Miguel has shared that we have had students with engineering backgrounds to our programs that do really well in our labs as well. So I just want to, don't self-exclude, just explore and ask us questions specifically to those areas. The next.
Yeah, can I add one really quick thing? So that's exactly right. We have students who are GSK students who have bioengineering, for example, undergraduate degrees. And something that comes up a lot is, well, what courses do I have to take as prerequisites to get into GSK? We have recommendations on the webpage. If you didn't take all the organic chemistry we recommend, for example, I would say please still apply. And you can say in your personal statement, I didn't take one of the courses that's recommended. That's okay, right? And also you can reach out to me to discuss if anyone has questions about that.
Thank you, Mike. I think this is in relation with the computational biology summer program. It's the student is asking, how is the computational biology program? Is it bioinformatics?
Yes. So there's more information about that on the web. If you look at it, I may not do it justice, but we have a whole computational biology department at Memorial Sloan Kettering. And then in our Tri-I community, which involves Weill Cornell and Rockefeller, there's a lot of computational biology that includes bioinformatics in our community. You might imagine we're actually, if you consider the Tri-I, but also MSK, we have a powerhouse of bioinformatics, and in particular with clinical-driven data sets, which Adrienne could speak to better than I could. We sequence every patient that comes through. So there is an enormous need for computational expertise and dedicated laboratories for that, and lots of collaborations everywhere. So check out the summer program we have in computational biology. There's also a tri-institutional PhD program in computational biology that you can check out and find on the web. If anyone has follow-up questions, again, reach out to me. I'll shoot you an email.
Perfect. I think those are all the questions that we received in the chat. We have 2 minutes, so if there is any pending question, I will be happy to read them. Mike, maybe you could type in your email or the GSK email in the chat.
Definitely.
We receive questions. And I want to plug one event that we are hosting next week. Under GSK, we're going to be hosting an information session about how to apply for grad school, and we're going to be covering all the bases from applications to interview. It's open, it's free. I hope to see as many of you at this session as you can. I will share the registration tiny URL in the chat box. But the idea, this is not a recruitment strategy for us. It's actually like, I don't want to be sitting in all this knowledge, and I want to help this generation get into grad school. You guys have endured a pretty unique year, and we want to be here for you, helping you and providing you advice on how to apply and put out strong applications for grad school in biomedical fields. Not only to us, hopefully you come to us, but if you decide to go elsewhere, we want to give you the tools to be competitive and put forward the best application. Yes? We don't have many more questions, Carmen. I think they're a little bit shy with me.
Can I give one piece of advice? Because it's in response to a question we get quite a bit, but not yet today, which is, what about research experience? And in the letter, what should I write about? And I want to make one point. Anyone else can chime in if they want, but in particular, we just went through COVID. We're going through COVID, and there are other experiences that can limit research opportunities. And I want to give one piece of advice. It's just as important when you write your statement, how you speak, how you write about the research experience you do have, as how many experiences you have had. And I think what I hear too often with students these days is, well, I don't have enough research experiences. That may not be true, actually. It may come down to how you write about the experiences that you have had. And so I would really encourage all of you to apply. Particularly, we have no fee, right? So, apply. Or if you want to discuss, reach out and chat with any of us about it. But that's something we get all the time. I don't think I've had enough research experience. And while that can be the case sometimes, focus on the experience you had and how you write about it to other scientists, and that will take you very far.
Thank you, Mike. And I think at a personal level, I am a product of the University of Puerto Rico, the same as Miguel and Marimar, and I wouldn't be here today if it wasn't for the training that I received there. So I speak volumes, and I am a big advocate of University of Puerto Rico students. So I really hope that you take this chance to submit applications, but moreover, reach out to us and let us know how can we help you in the process. Thank you. And Carmen, all yours.
Thank you all for this wonderful opportunity for our students to interact with you all and to know more about your programs, which I already know a couple of my students texting me like, I want to apply. So you will have some of us applying for sure. And thank you, Adrienne, so much for being here and sharing your science. That is so groundbreaking, and I really appreciate to have the opportunity to meet you and know that there's a possibility maybe one of our students will go to your lab, and that would be wonderful. And among the others, of course, wonderful scientists that Sloan Kettering has for us. So I have to say thank you and finish our activity because I want to finish on time. And once again, thank you all for being here. And I encourage you, if you want to come back next week on Wednesday, we will have our next presentation, and hopefully we all will have such a wonderful time as we did today. So thank you all. Gracias mil a Marimar y a Miguel, y por supuesto a Yaihara. Gracias a todos. Thank you
for the opportunity. Thank you.
Thank you.
Thank you.
Take care.
Bye.